Hello everyone, welcome to Modern Biotech YouTube channel. Today we are going to discuss about what is mRNA vaccines and why it is called as a breakthrough in vaccine productions. So let's get started. So in the recent time it got limelight due to the Nobel Prize won by two scientists for the research in mRNA technologies that gave us a basic principle and tell us how that mRNA vaccines generate an immune response in our body. And also, the mRNA vaccine came into act firstly in 2020 when that first vaccine against COVID-19 came into the act, and we can see that how that if vaccine is effective against the COVID-19 and how quickly that our scientists generate a vaccine against the COVID-19 that tell us that mRNA vaccine is a very easy and fast way that we are going to see in our upcoming slide also. And before starting this video, first we need to understand what is mRNA. mRNA is a single standard RNA that encodes for the proteins that can be used for multiple purposes in our body, like structure proteins, functional proteins. That's why it is called as, uh, in simple word, if we say, it's like any protein which is encoded in our body or produced in our body is indirectly or directly responsible for the function in our body. That's why it is also called as functional unit of life or body. Okay, and if we see the history of that mRNA vaccines, so in 1960s mRNA has been discovered, and Francis Jacob and Jacob Monod got a Nobel Prize for this discovery in 1965. Then in later 2000s and 2003, the Peter Cullis developed a lipid nanoparticle because as we know that mRNA vaccine is a very effective vaccine. But if we don't have a particular delivery system for delivering that mRNA vaccine, nothing will happen in our body, because we know in our body RNAs and DNAs, multiple proteins are there, multiple enzymes are there, and due to the chemical arrangement of in our body also, if we insert that mRNA vaccine due to our oral or without any particular delivery system, it can degrade easily. That's why the effect that mRNA vaccine can cause it in our body can be neglected. That's why the important is that we need to find a particular method by which we can deliver that vaccine into our body. And then in later 2006 or 2007, Kathleen Karaoke and Drew Westmans were introduced the mRNA engineering concept, which tell us that what how that cell can trigger the body defense mechanism using the mRNA technology. This is like a revolutionized mm, research, because after this they gave the basic principle that if we can also use the mRNA as a vaccine, because like in traditional way, if we want to develop a vaccine against any pathogens, what we need to do, just we need to take we can try the inactive vaccination, we can try the subunit vaccine, and we can try that whole cell vaccine. This is the that we can use for traditional vaccines, but it has some pros also, cons also. So that we are going to see in upcoming slides also, why that mRNA is better than the traditional vaccinations. Okay, and then as we discussed earlier also, that first mRNA vaccines came into act in 2020 for the COVID-19. Then what is mRNA vaccine? As name suggests, vaccine uses mRNA. mRNA is like a messenger RNA that is that encode for the spike protein of that virus, that will be inserted in our body, and then it will go into our cells, and the cell will recognize that mRNA and produce a spike protein that will utilized by, or that will recognized by our immune cells, and immune cell as an antigen. Okay, and it will be degraded by them, and generate immune response will be generated. So what is the benefit in this one is, as much as that mRNA present in our body, it will be encoded and spike protein will be generated, and the immune response against that specific pathogens will be long lasting. This is the importance over the traditional vaccines. Okay, and if you see the rise of the mRNA vaccines, so as I told earlier also, the proper way of delivering that mRNA vaccine is an important task. So pupils are trying a multiple ways like lipid or solid nanoparticles, and novel applications are also discovered. Okay, by which we can insert that vaccine in mucosal delivery. By means through our oral vaccine we can develop, or by generally these vaccines are inserted into that arm area, and in by which it will be released into our cells, and spike protein can be generated and immune response can be generated. And these are multivalent and therapeutic. It can be used as a multivalent and therapeutic cancer vaccines also. And the importance of this vaccine is it generates a long lasting humoral and cellular immune response, and a low amount of dose is required for immunization. Okay, if we see the advantage, and in advantage, firstly, if we see about the safety, so it is too much safe because it is because we are inserting into the body is just mRNA, not any pathogen, specific pathogens. Okay, that's why it is non-infectious, and also this is mRNA which is not going to insert in your DNA. That's why DNA integration also can be overcome, and this is cell independent process for the production of vaccine. That's why it's toxic chemical agent free. That's why it is safe for the body of the human beings or any patients. Okay, and if we talk about the efficacy, because we are using a mRNA packed into lipid molecules, this will increase the efficiency of our vaccines. Okay, and also as much as I told earlier, ki mRNA is present in our body, it will encode for spike protein, and that will generate an immune response against that pathogens. Okay, and if we talk about the productions, it is very easy to produce, and it can be easily scale up also, and this is a sequence independent process. So what just we need to do, just take a pathogen, find the spike protein, isolate the mRNA, produce in large quantity, and just use it as a vaccine and insert it into the human beings. Okay, and it is very low cost, and as much as that mRNA vaccine technology will develop, that vaccine cost will also be decreased in the future purposes. If we see the advantage over that mRNA vaccine over a traditional vaccine is just, if we see the comparison only, that component of mRNA vaccine is mRNA, and component of traditional vaccine is, it may be microbial protein or it may be the inactive microbe. Okay, and if we talk about the production, how it can be produced, the mRNA vaccine, it can be produced by just mRNA molecules and easier to produce. Okay, and production of traditional vaccine is just slower and more difficult to produce because we need to grow that microbe, and if we are going for whole cell vaccination. But if we are going for subunit vaccination, what we need to do, we need to isolate that protein, we need to produce in vitro conditions like in lab using RDNA technology, and then we need to isolate that protein. That is a very hectic task, or it very difficult task also to produce, to get a specific type of proteins. Okay, that's why it's very time consuming process compared to mRNA vaccine. And if we talk about the process, the components are injected into the arm and serve as the instructions for the body to make a microbial proteins. The same thing which I explained earlier also. And also if we talk about the traditional vaccine procedures, the same, like we are inserting a vaccine in the form of whole cells or microbial protein that will be used by, that will be recognized by our immune cells like B cells, T cells, and different types of cells that are playing a important role in immune system, and then it will generate a specific type of antibodies against them, and immune response will be generated, and our body will be ready to fight against that pathogens. Okay, these are the two heroes who plays a very important role in the development of mRNA vaccines, because as we see, Dr. Kathleen Carrique is both together work too much and develop a technology that can be used, that can be like, that can decrease the production of vaccine, like that can make it easier way to produce a vaccine against any pathogens. Like if we go in traditional way, what we need to do, if any pathogen, if any disease is there, what we need to do, just isolate that pathogen first, then identify the antigenic proteins, then uh, see which type of vaccine is more suitable for that one, like subunit vaccine or whole cell vaccine, then produce a vaccine against that, and check, then go for the clinical trials and preclinical trials, and then go for the main injection. That will take like 4 to 5 years. But if we see that in case of mRNA vaccines, what we need to do just in viral case, like we need to identify the spike protein mRNA, then take that mRNA and do the preclinical trials and just insert into the body and generate a infection against that pathogens, by which that pandemic situation which is created outside or due to some infections, it can be overcome. It can be treated very quickly. That is a importance of this mRNA technology which is given by these two scientists. Okay. Now, after these all things, one question will be coming in your mind: what is next for the mRNA vaccine? Okay. Now, in the future prospects, mRNA-based vaccines is going to fill the gap between emerging pandemic, infectious disease, and bountiful supply for the effective vaccines. And also, the clinical and preclinical trials are going on to develop a long-lasting immune response in animal model and humans model, by which we can generate too much long-last immune response. Okay, and also the different methods are trying to develop for the delivery of these mRNA vaccines, by which it can be easily it can be easily go into the target, and that infection can be prohibited, or that immunity can be generated. Okay. In future, if we talk, that mechanism of action and potency are still needed for the full development of mRNA vaccine. Means this technology is in under development. So it need to develop more. So as much as the research will go into this mRNA vaccine, it will develop too much, and in future, if any pandemic is coming, like in COVID-19 situation, we can easily overcome that pandemic, and the loss which we got during the pandemic in case of our human beings, it can be reduced. That is a basic importance of this mRNA vaccine technology. Then there is one scientist, Nicholas Wayne, who is a vice president of pharmaceutical R&D at Pfizer. He says about that mRNA vaccine that if we look forward and really let our imagination go and start marrying unmet medical needs to this new toolkits, the toolkit is mRNA technology that we have developed over the past couple of years. It is going to be really exciting. Means if we develop in this mRNA technology, it will be going to be revolutionized. It will be very helpful in medical industries. And for for now, for this video, we are going to end on this slide. And thanks for your attention. I know that from past few months I didn't posted some videos, but from now onwards I will try to post at least one video in a 1 week. I am also in middle of something that you already knows. I am doing the MTech, so I will try to post regular videos. And just if you have any doubt regarding this video, just comment down, and I will try to solve that issues. And also, if you want any type of particular videos, you can also tell in comment section. Okay. Thank you. Thank you for this. Thank you for your attention. We will meet in next video. Thank you.